Hello everyone. So today we're going to continue 7.3. This is going to be the last topic within 7.3. So we're going to talk about protein destination and protein structure. So one of the first key understandings that you must remember is that there are different types of ribosomes. So there are free ribosomes, and these are ribosomes that are in the cytoplasm of the cell. And the proteins that they make are usually going to stay within the cell. That means that they're not going to be exported out of the cell. And then there's bound ribosomes. So these are the ribosomes that are attached to the ER, and they're going to be primarily used for secretion. So that means that they are going to be exported out of the cell via the rough endoplasmic reticulum, Golgi apparatus, and then vesicles. Or they can also be used in lysosomes that are also other organelles within the cell. So that's the first essential understanding and differentiation that you need to make. So remember that all proteins produced by eukaryotic cells are initially synthesized by ribosomes that are found freely. So that means that these bound ribosomes are not bound initially. They become bound because of a process that you're going to see in a minute. So all ribosomes start as free ribosomes that are circulating in the cytosol. Okay, and then what I said, right? That if they make proteins that are to stay within the cell, so intracellular proteins, and the ribosomes remain free and unattached. If the protein is targeted for secretion, membrane fixation, or in the use of lysosomes, then the ribosome binds itself to the ER, and that's what we refer to the rough ER, the rough endoplasmic reticulum. Okay, so how does a ribosome know whether to stay free or whether to go attach itself to the ER? Well, all of this is determined by the protein itself. The protein itself carries a initial signal sequence. So the nascent polypeptide chain, that means it's the protein that is just being made, carries an initial sequence, and then that's going to target a biochemical cascade that is going to direct the ribosome to the right place, right? So either stay free or go attach yourself to the ER. So this is the process that we're going to study right now in a little bit more detail, how this protein uses this initial sequence to drive the ribosome to its correct location. So I'm going to scoot this up a little bit so you can see here. So the first thing we need to understand is that right here we're looking at the ribosome and the nascent polypeptide, or the growing protein, that's found within the cytoplasm of the cell. Okay, so this ribosome happens to be free floating. And then right here they're showing us the phospholipid bilayer, or the membrane that makes up the endoplasmic reticulum. So here we see the inside of the endoplasmic reticulum, also known as the ER lumen. Okay. So here we see the ribosome with its two components. mRNA has been translocated, and we see that translation is happening. So we see that the polypeptide is starting to grow. So this polypeptide releases a signal sequence. Deep, deep, deep. Yeah, sound effects and everything. So the signal sequence, that results in a recruitment of another particle. And this other particle happens to be a protein also. So this other particle is called an SRP, or signal recognition particle. The signal recognition particle then binds to the protein who is releasing that signal, and it forms a complex. So now we have an SRP ribosome complex. So this is what's going to direct the ribosome to go and dock on the membrane of the ER. Okay, so once the complex here docks, it actually attaches itself to another protein within the ribosome. within the ER. Okay, so it docks at a receptor located on the ER membrane. So now this is what we're able to see as the rough ER, right? Because these ribosomes are docked here. All right. So what's going to happen? So the ribosome is going to continue the translation process. It's going to continue synthesizing the protein. And now this protein, it's going to get eventually transported from the ER via vesicle to the Golgi, and then via vesicle again for secretion, or via a vesicle to the lysosome. Okay, but also remember that this membrane within the ER, it also needs proteins itself, right? So here you need a protein for this docking process. Here you need a protein for the new protein that's being made to actually get inside the ER. So some of these proteins are going to become integral proteins. That means that they're going to be used also to get embedded in the ER and form part of the ER membrane. So that's something else to remember. So let me move this a little bit further down, and you can see that after the SRP and the protein complex attaches to receptors on the ER, the translation process continues, right? And then what happens? Well, the signal sequence at the end has to be stopped, right? And has to be cleaved. So at some point here, there's got to be a stop codon that's reached. Synthesis stops. The signal stops. It's then cleaved. And the SRP, which is this molecule here, or protein, is recycled. So it can be used again. So here you have it. Here you have the protein that is being synthesized into the ER lumen. Okay, so again, these are going to be proteins that are going to be exported, right? If they are bound to the ribosome, they're going to be exported. So this picture here is not showing us what's going to happen to the proteins that are made and that stay within the cell itself. So this process only shows us what's going to happen to the proteins that are made after the docking of the SRP ribosome complex to the ER membrane, right? Because again, these proteins are going to be exported in some way. Okay? Okay. So that's the first part, right? Protein destinations. So now let's look at protein structure. So here we see that proteins have different levels of protein structure. So we have a primary level, secondary level, tertiary level, and quaternary level. Okay, so we're going to study a little bit what these structures look like. So the essential understanding for the primary structure is the sequence and number of amino acids in a polypeptide is the primary structure. So here we see these orange circles. So these orange circles are amino acids, and amino acids, if you remember back from topic 2, they have a particular biochemical structure. On one end, they have this functional group called an amine, which is NH3, and on the other end, they have another functional group called a carboxyl group, COOH. These functional groups are sometimes referred to as R groups because they vary, right, within the protein itself. And we're going to see other R groups in just a moment. The other important thing about this is that these amino acids are held together by peptide bonds. So here we see the peptide bonds hold the chain of amino acids together, and that's the reason why proteins can also be called polypeptide chains, right? A chain of amino acids. All right. Even though it's not being shown here, we know that one amino acid has an amine group on one end, and that's bonding to the carboxyl group of the other end of the other amino acid. Okay, And that gives us a simple primary structure. And this is going to be important because even though it has no functionality, that's something important to remember about primary structure, it has no functionality, it's actually going to determine the other subsequent levels of protein structure. Okay? So that takes us to our secondary structure, and secondary structure is it's a little bit prettier. So the secondary structure is the formation of these alpha helixes, and you've heard me talk about these before, and these beta pleated sheets. And the reason why they are able to form in this way is because they're stabilized by hydrogen bonds. So here we see an alpha helix right here. And notice that these alpha helixes, they look like little spirals like this in purple. And here we have the beta pleated sheets. And sometimes beta pleated sheets can also be represented as arrows. Okay, so these blue arrows here. So what happens? How does a primary structure turn into a secondary structure? Well, the amino acids fold. And when they fold, they form these repeated arrangements of alpha helixes and beta pleated sheets. And the folding is a result of the hydrogen bonding. So here you see the hydrogen bond right here between the amine group of one amino acid and the carboxyl group of the other amino acid. So here's the amine group and here's the carboxyl group. So depending on where these hydrogen bonds form, then you're either going to get your alpha helix or your beta pleated sheet. So here we also see the amine group right here and the carboxyl group right here, and then we see the hydrogen bond here in the middle. And keep in mind that sometimes you have sequences that do not form either alpha helixes or beta pleated sheets, and they're just going to exist as a random coil. Okay, so this hydrogen bonding that just happened here, this allowed for some type of mechanical stability. So this means that proteins that are of secondary structure are usually found to have a function of structural support. So we know that if you are a fibrous protein, and you learned this back in standard level, if you are a fibrous protein, like keratin in your hair, collagen in your skin, then you have a fibrous nature and you're there to provide a structural support. And we see that, for example, that same collagen and keratin, they are made up of these alpha helixes and beta pleated sheets. Okay? All right, moving on. Then we have the tertiary structure. And the tertiary structure, I like to think of it as a three-dimensional shape. So that means that you get even further folding of the polypeptide, and you get further interactions between R groups. So this means that instead of just having the amine groups and the carboxyl groups, there are other groups there that interact. And when they interact, they stabilize the three-dimensional shape of this tertiary structure. Okay? Now, this is the reason why these tertiary structures are called globular proteins. And since they're globular proteins, they're catalytic, so they have a catalytic function. So, for example, all enzymes are tertiary structures like this. Okay, they're globular and they're tertiary. So let's look at some of the interactions between the R groups. So here we see a hydrogen bond. Hydrogen bond. Here we see a hydrogen bond interaction. So here you have one interaction here between one R group here and another R group here. And you're not required to know all the names of these R groups. Okay. And then we see a disulfide bridge here between an R group here and another R group here. So this causes certain parts of the primary structure to continue folding. And then we see some ionic bonds here between R groups here and R groups here. And then we even see some hydrophobic interactions here and here, right? So then that means that you're going to end up with a convoluted type of three-dimensional structure. Okay. They also mentioned that the relative amino acid position is important. So remember that all of this is dictated by the DNA, right? If this is a chain of amino acids, well, it's a chain of amino acids with the particular sequence of amino acids is dictated by the DNA. So if you end up with a chain of amino acids that's mostly nonpolar, then you are going to end up with a protein that avoids exposure to aqueous solutions. All right, so it's not going to be very compatible with water. What else? What else? Yes, tertiary structures may be important for the function of the protein. We do know it has a catalytic function. So it's important because if we don't get these tertiary structures to fold properly, then that could be disastrous if you're an enzyme, right? Because if you're an enzyme, you need to have a very specific active site. Otherwise, the substrate's not going to be able to bind. So when these tertiary structures are folding, right, that sequence of amino acids, as dictated by the DNA, needs to be very specific, and it needs to be folded correctly. Otherwise, the enzyme is not going to work. And sometimes we see defects in the genetic code. So these are genetic conditions, which means that a certain protein of a catalytic nature is not going to be working properly. All right, and then finally we have the quaternary structure, and this is easy to spot. Oops. This is easy to spot because you actually see more than one tertiary structure. So if we recognize this as a tertiary structure, let me move this up a little bit more. So if we recognize this as one tertiary structure, well, if we put 4 of them together, 1, 2, 3, 4, then we have a quaternary structure. So this tertiary structure, remember, and here is drawn in different colors for 2 reasons, because remember that this tertiary structure can have a different sequence of amino acids, right? And also because some of these tertiary structures carry these prosthetic groups, okay? So here we see a prosthetic group here and a prosthetic group here. And a prosthetic group, what it is a biologically active protein, okay? When the prosthetic group attaches to the protein, it forms a biologically active protein, which is your quaternary structure. The prosthetic group is an inorganic compound. So, for example, in hemoglobin, we know that the prosthetic group inside hemoglobin is the heme group. Now, the heme group is made up of iron, right? Because we know that inside our hemoglobin we have iron, right? So it could be +2, +3, who knows? So the heme group here is the inorganic compound or the prosthetic group attached to this tertiary structure, right? And for example, in hemoglobin, the prosthetic group is iron, okay? So now if you get all of these tertiary structures together with their prosthetic groups, you end up with a quaternary structure, okay? And they are held together by all types of variety of bonds that are very similar to the bonds that we just studied in a tertiary structure. So we know that hemoglobin has a function, right? It has a function to carry oxygen. So we know that quaternary structures are also catalytic in nature. Okay, so that is it for today. Let me know if you have any further questions in class.